spindle cell tumor of soft tissue. I will concentrate on histomorphology only. 7 points should be considered before reporting. Are the tumor cells forming any pattern? Are they in seeds or fascicles? If in fascicles, what type of pattern is there? Zonal pattern, that is hyper and hypocellular areas, herringbone, storiform, multinodular, reticular or microcystic, crisscross, checkerboard pattern. Are the fascicles divergent? Cytologically, one should see for clear cells, granular cells, rhabdoid cells, epithelioid cells, nuclear palisading, and pleomorphism. Is the tumor mitotically active? Is the stroma collagenous, or collagen rosettes are present or not? Collagen wiry or ropey? Vascularity, chicken wire type or staghorn type, which is also called hemangiopericystoma like vasculature. Infiltration is present or not? These are the conditions I will discuss. For the sake of simplification, I have divided into 3 groups. This is the first group. In case of nodular fasciitis, irregular fascicular growth pattern, which is also called tissue culture like. There may be zonal pattern also, that is hyper and hypocellular areas, bland chromatin, frequent mitosis but no atypia, microcyst may be present, infiltration absent, and collagen entrapment absent. In case of cellular dermatofibroma, storiform pattern, focally nuclear pleomorphism and prominent nucleoli. There may be infiltration in the subcutaneous fat around lobules, but there is no honeycomb type of infiltration. Here in this case, we will find collagen entrapment at the periphery. Solitary fibrous tumor, bland nuclei, tumor is not mitotically active, stored on vessels, perivascular hyalinization. If the mitosis is more than 4 per 10 high power field, nuclear atypia and necrosis, one should think of the malignant counterpart of solitary fibrous tumor. In case of desmoid tumor, long divergent fascicles, bland nuclei, and entrapment of skeletal muscle fiber at the periphery. In case of dermatofibrosarcoma protuberans, storiform pattern, no microcyst, no inflammatory cells, honeycomb type of infiltration around individual adipocytes. There is no collagen entrapment. This is the entrapment of collagen. Collagen bundles are surrounded by fibroblast. This is honeycomb type of infiltration found in cases of dermatofibrosarcoma protuberans. Here you find tumor infiltrating around individual adipocytes. This is the second group. In the second group, monofacial synovial sarcoma, fibrosarcomatous appearance, bland nuclei. Nuclei are very close to each other, appears to be overlapping one another, wiry collagen, perivascular hyalinization, and staghorn type of vasculature. Infiltration is to be present. In case of leiomyosarcoma, fascicles are at right angle, cigar shaped nuclei, that is blunt rounded ends, focal pleomorphism, tumor is mitotically active, and staghorn type of vasculature. In case of spindle cell rhabdomyosarcoma, it has got diverse pattern like lobules, nests. One can get rhabdomyoblast, tumor is mitotically active. There used to be eosinophilic and basophilic matrix. In case of cellular schwannoma, fascicular growth pattern, cells narrow, elongated, and wavy. Tumor is not mitotically active, hyalinized vessels. In case of malignant peripheral nerve sheath tumor, fascicular growth pattern, as well as marble appearance because of hyper and hypocellular areas. Hyperchromatic nuclei, thin, wavy, buckled. A few epithelioid cells may be seen. Tumor is mitotically active. There is accentuation of perivascular cellularity. In case of clear cell sarcoma of soft tissue, multiple patterns used to be seen, like nested, school of fish, diffuse, alveolar. A few epithelioid cells may be seen, as well as clear cells. There used to be inflammation and cystic degeneration. This is monophasic synovial sarcoma. Nuclei are crowded, appears to be overlapping each other. This is leiomyosarcoma. Fascicles are at right angle with each other. There is nuclear pleomorphism and a few giant cells. Rhabdomyoblast in cases of spindle cell rhabdomyosarcoma. Cigar-shaped nuclei, blunt rounded ends, and pink cytoplasm. This is cellular schwannoma. Narrow elongated wavy cells with tapered ends. Malignant peripheral nerve sheath tumor. Hyperchromatic thin nuclei. Clear cell sarcoma. Short vesicles, appears to be like a school of fish. This is the third group. In case of spindle cell lipoma, bland nuclei, a few florid giant cells may be seen, and scattered lipoblast without atypia. Mitosis is not increased. Tropy collagen. In case of perineurioma, storiform pattern, wavy or tapering nuclei, long cytoplasmic processes, degenerative atypia may be seen. In case of low-grade fibromyxoid sarcoma, swirling or hold pattern, bland spindle cell or sterile cells, a few epithelioid cells may be seen. Tumor is not mitotically active, but we will find infiltration and collagen rosettes. In case of low-grade myofibroblastic sarcoma, storiform pattern, focal moderate nuclear atypia with hyperchromasia and irregular nuclear membrane. Small nuclei may be seen. Tumor is not so mitotically active, but we will find infiltration. This is florid giant cells. Hyperchromatic nuclei, wreath-like arrangement, pink cytoplasm. This is atypical lipoblast in cases of liposarcoma. Well-defined vacuoles, nuclear indentation and hyperchromatic nuclei. This is perineuroma. Wavy tapering nuclei, lining cytoplasm, long cytoplasmic processes. This is collagen rosettes, found in cases of fibromyxoid sarcoma. One should always exclude carcinoma, melanoma, lymphoma, mesothelioma, just in case of abdominal cavity. Role of immunohistochemistry and molecular studies are significant because of various morphological patterns. Thank you.